Good afternoon, people. I am Soh Jun Xing from PA2 and today I'll be going through the pre-lab video for sample preparation based on a journal article. The study's main aim is the determination of metformin in human plasma and urine by high-performance liquid chromatography using small sample volume and conventional octadisylsilane column. For those of you who do not know, metformin belongs to a class of drugs called liguanide and is used to treat diabetes. The sample preparation method, as described in the study, is liquid-liquid extraction or LLE. The reason why we are using LLE is because the samples we are dealing with here are human plasma and urine, and these are both considered aqueous samples. In order to separate the constituents from the aqueous samples, we will then need to add another immiscible liquid, which then makes use of the solubility of the analytes, or in this case, metformin, to dissolve in the different solvents. The first step is to prepare the internal standards. For plasma, it will be 100 microliters of matrix to 20 microliters of internal standard, and for urine, it will be 100 microliters of matrix to 30 microliters of internal standard. This will be your reference for HPLC. Second step is to add 80 microliters of 10 moles of sodium hydroxide to the media in order to alkalize it. Sodium hydroxide is chosen because it is a very strong base, and since metformin is also a very strong base, metformin will be in unionized form or non-polar form in the alkaline pH. Third step, you'll need to add a 3 ml mixture of 1-butanol and hexane at a 50-50 volume by volume into the aqueous sample. Both 1-butanol and hexane are non-polar compounds and are organic. This mixture will be immiscible to plasma or urine, which is crucial for LLE as the analytes will prefer the hydrophobic environment of the organic solvent. Or to simply put it, like dissolves like, so the non-polar analytes will be attracted to the non-polar solvent. The fourth step is to vortex mix the tubes for 30 seconds, followed by centrifuging at 3000 force for 3 minutes. This step is to ensure that there is maximum mixing between the aqueous and organic layer, which then allows the analytes to move from one layer to the other. After centrifuging, you should be able to see an organic layer, or what we call supernatant, at the top of the tube. The analytes of interest should be in the organic layer now. You are then required to transfer it into clean glass tubes and acidify the supernatant with 500 microliters of 0.2 acetic acid. The reason we add acid after retrieving the organic layer is because we want to remove any ionic inorganic material that might contaminate the contents in the tubes. After you are done acidifying, vortex mix the tubes for 60 seconds, followed by centrifuging at 3000 force for 3 minutes again. This is again to ensure maximum mixing between two layers so materials can move from one to the other. After centrifuging for the second time, you will then carry out aspiration using a pipette attached to a vacuum flask. This is to remove the supernatant from the aqueous layer. The remaining aqueous layer will then need to be evaporated into dryness in a vacuum. We do evaporation in a vacuum because we want it to be fast, but also avoid using heat, which could potentially destroy our analytes of interest. For the last step, you'll need to add 200 microliters of HPLC water along with the residues for reconstitution. We reconstitute the residue so it will be suitable for HPLC analysis. Remember to set the injection volume range to be from 60 to 70 microliters on the chromatographic system. This is the resources used for this presentation. Thank you for watching.